In our second segment, we're going to talk with Dr. Boynton about functional magnetic resonance imaging. Could you tell us about what that is, how it works, and what it measures?
Sure. fMRI is a way of measuring brain activity in the human brain without using any sort of invasive technique.
Mm-hmm.
And what it's used is a traditional MRI scanner that is basically tuned to pick up on changes in the blood flow and oxygenation that's associated with brain activity.
Mm-hmm.
So normally if you go in and take an MRI of your knee or your brain or something like that, it's designed to take an image of the brain, a high resolution spatial image of the brain that you've probably seen in pictures.
Mm-hmm.
Functional MRI is actually picking up on not on the structure of the brain, but on changes in the intensity of the images that are associated with blood flow and oxygenation that flow around in association with changes in neural activity.
Mm-hmm. So neural activity is coupled with blood flow.
Mm-hmm. Yeah, so fMRI relies on actually two facts. One is this interesting fact that when there's a change in neural activity, say a subclump of neurons in the back of your head, there's an associated flow of oxygenated blood to that region of the brain.
Mm-hmm.
So it seems kind of backwards because you would think that when neural activity occurs, there's metabolic activity and there'd actually be a decrease in the oxygenation there. Just like when you move your legs and stuff, there's metabolic activity using up oxygen. But actually, and that does occur, but it turns out that the vasculature system of the brain changes in such a way that it sends more oxygenated blood to the active part of the brain.
Why does it do that?
We don't really know. We think that it has something to do with the best way to get blood or oxygen to neurons that have been starved because of metabolic activity is to actually increase the oxygenation content so that the oxygen will diffuse through the tissue to get to these neurons.
Is that a solid assumption to make, that neural activity is associated with the metabolic functions of neurons?
Well, it's all we've got. And so it's been over 10 years, about 15 years now since fMRI was originally developed. And it's based entirely on this link between neural activity and blood flow changes. But still surprisingly little is known about this link. We hope that there is a direct link between a neuron firing, having spikes, and a change in the oxygenation to that location, but we really don't know. We don't know what's causing this.
What does the activity represent? Is it actually the neuronal firing, or is it synaptic activity?
Really good question. So both of these things involve metabolic activity, neuron firing, synaptic activity. We hope it's firing because that's what the monkey physiologists are measuring, so that makes us hopefully measuring something that's very similar. But if it's synaptic activity, it could be a real problem. Because as you know, there's inhibitory synaptic activity that's actually more inhibitory connections than excitatory connections. Mm-hmm. If an inhibitory connection leads to an increase in blood oxygenation, then we've got the sign wrong. That means more inhibitory synaptic activity can actually reduce the neural firing rate but increase our blood flow signal. That'd be a big problem.
So what was the second thing that makes fMRI work?
So the first thing is this link between neural activity and blood flow changes. And the second thing is how to use an MRI scanner to detect where the blood oxygenation is going. And this relies on this fact that oxygenated blood, red blood, and deoxygenated blood, blue blood, have different magnetic properties. And specifically, blue blood, deoxygenated blood, is paramagnetic. It acts like a little dipole in itself that will screw up the local magnetic field. And normally with MRI that's a problem because it causes the signal to be weak where there's any kind of magnetic material in there. But what it does for us is it means that our images are slightly brighter where there's more oxygenated blood and slightly dimmer where there's deoxygenated blood. And so just as a result, where there's more oxygenated blood, we happen to have slightly brighter images.
Can the magnetic field emitted from the fMRI change the vasculature at all?
Yeah, we don't want this kind of Schrodinger problem where the process of measuring actually influences the brain responses. So far, we don't think that that's happening. It is true that a rapid change in the magnetic field, which is what we're doing with MRI, can induce neural activity. And in fact, I've experienced this when I'm lying in the scanner doing an anatomical scan. You can experience something called peripheral nerve stimulation, where you might feel a muscle twitch every time the image is being acquired. But for functional MRI, the gradients or the changes in the magnetic field that we're using are not fast enough to induce neural activity, as far as we know.
So You're talking about how fast it is. What is the temporal resolution of fMRI? And while we're on the subject, what is the spatial resolution?
So the biggest weakness of fMRI is its very poor temporal resolution. And people need to understand this when they're reading like newspaper articles about fMRI. We're not measuring neural activity directly. We're measuring these changes in blood flow that are quite indirect and quite slow. So, for example, if you take a sequences of images over time with fMRI and measure the time course of the response to, say, an onset of a visual stimulus. So if I flash a visual stimulus, nothing will change in these images for a couple of seconds. And then the response will slowly rise and peak after about 5 or 6 seconds and then decay. And the whole thing won't end for like 15, maybe 20 seconds. So it's very, very slow. Neural activity is in the order of milliseconds. We're measuring things in the order of seconds, which is way different. Spatial resolution is much better. It's in the order of millimeters. So high resolution fMRI done in the highest field scanners, like the ones we have at the University of Washington, with the right kind of receiving equipment, can get down to about a millimeter of resolution. 1 by, say, 4 millimeters is a given, what we call a voxel, a pixel in our images.
What sort of techniques can be used to improve the spatial resolution and the temporal resolution?
So far, it's been pretty much brute force. In the early days of fMRI, it was done at clinical strength scanners, measured in Tesla, so 1.5 Tesla scanner is the kind you've probably been in if you've ever gone to the hospital and had a knee scanned. Research scanners now are up to about 3 Tesla, which is pretty standard. Stronger the magnet, the better signal we have. So better signal means that we can get smaller voxels, smaller pixels.
Do stronger magnets cause problems at all?
At some point, there's going to be issues. So there's human fMRI scanners that are now being built that are 7 Tesla. And so far, so good. But it turns out that you can actually induce, like I said, neural activity with big changes in magnetic fields. So eventually, we're going to run to some sort of physiological limit to how quickly we can change these fields in these scanners. Mm-hmm. They're also very difficult to work with. The higher the field strength, the more difficult it is to calibrate these machines to get a uniform field, and the more and more precise equipment we need to have for what's called the amplifiers and the gradients and things.
And so that's only improving the spatial resolution, or does that also improve the temporal resolution?
Really, the temporal resolution, there's not much we can do about with our current techniques because we're limited with these blood flow changes. There are efforts to try to measure neural activity directly with MRI. It is true that a change in electrical activity can induce a change in magnetic field, right? And that should be able to be picked up with an MRI machine. And so theoretically, with a high enough signal to noise, a strong enough magnet, and all these sorts of things, we might actually be able to get measures of neural activity more directly. But right now, the signal to noise is below our threshold.
Would it be easier to use event-related potentials to study the temporal dynamics of anything?
So EEG and its sister MEG, which is magnetoencephalography, are sort of the opposite of fMRI, where these have very, very good temporal resolution, but relatively poor spatial resolution. These are when you put electrodes on the surface of the scalp, and you're either picking up changes in the electrical signal or the magnetic field on the surface of the head that's induced by big bundles of neurons inside the brain. These measurements are instantaneous. As soon as there's a neural activity, there's going to be this change that we can measure. So we're really limited by the technique, the ability to measure in time. Mm-hmm. But spatially, it's very difficult to localize where these signals are coming from.
But when you use them both in conjunction, that's when you can look at both the accurate temporal dynamics as well as the spatial pattern that's elicited. So
there's more and more effort to try to do these techniques at the same time, to try to take advantage of both the spatial resolution of fMRI and the temporal resolution of evoked potentials. It's difficult because you can't really put a lot of electronics inside an MRI machine for obvious reasons. Mm-hmm. And so you have to go through a lot of tricks and filters and things to try to get these things to work. But people are trying to do both fMRI and evoked potentials at the same time to get best of both worlds.
So could you describe how an fMRI experiment actually works? Run through all the steps with us?
Mm-hmm. Sure. Yeah, one thing to keep in mind is with fMRI is it's not like you put somebody in the MRI scanner, show them a stimulus, like a visual stimulus, take a picture, and then publish it. What fMRI relies on are differences between responses in different conditions. And the main reason is that our fMRI signal, like I said, is very, very slow, and it also kind of drifts around a lot. And so the absolute level, the intensity of an image that we get out of the scanner doesn't tell us much. It's really relative to other states. So typical fMRI experiments, like a classic called a block design experiment, involves alternating between two conditions. Say, like I said, attend to the left side of the visual field, attend to the right side of the visual field. And we look at the differences between those things. Mm-hmm. So in a typical experiment, a subject will lie in the scanner. They'll be looking at a mirror that is pointing either back over their head or towards the screen at their feet. We have a projector, an LCD projector that's projecting an image onto the screen that's synced up with the scanner so we know when we're presenting the images. And we'll simply have the scans last about maybe 3, 5, 7 minutes long, where we cycle through different stimulus conditions, like attend left, attend right, attend left, attend right. And during that time, we acquire these images, say every 2 seconds, during that whole thing. So we're basically taking a movie of the brain over time. And the next step is to compress this movie down into, say, a single data set. So what you can typically do is you look at the response of a given pixel over time, watch it go up and down, and then we want to turn that into a single number. So the amplitude of that modulation will define how well that pixel is responding to the visual stimulus.
Do you do that for every pixel in the brain for every experiment, or do you focus on specific regions of interest? Or?
Well, we get the whole brain. So basically we get this, the data set is a volume that we take every 2 seconds over time. So a typical fMRI data set will have, say, 32 slices that are maybe 3 millimeters thick, and each slice will have 64 by 64 pixels in there. And so we get this box of data every 2 seconds. And then, like you said, we basically look at the response of every single voxel, even the ones that are outside the head, and analyze those in sync with the stimulus or the task that we gave the subject.
So it's all set up on an XYZ coordinate system.
Mm-hmm.
How do you actually know which voxel corresponds with which brain region?
That's an important issue with fMRI. Is, okay, now we've got this voxel that is responding strongly to our stimulus or our task. Well, where is it? So what we do also in a typical fMRI session, either at the beginning or the end, is without moving the subject's head, we take a very high resolution anatomical scan. It's called a T1 weighted scan, which is, like I said before, it's a traditional way of using the scanner, which will give you a picture of the brain that actually looks kind of like an X-ray. Mm-hmm. And we align our functional data with that thing right there. So if I've got a voxel that seems to be responding strongly to the stimulus, I can look at where it is in terms of the anatomy of the brain there. That's the first step. And of course, great. So now I know that there's a voxel in the back of my head in the gray matter right here that responds to a visual stimulus. Now what? How do I know where, in terms of functional areas, that voxel is? And so the next step is to try to break the brain down into different areas.
You mean how to functionally define the different regions?
Exactly. So, and visual system is relatively easy because there are visual areas that are adjacent to each other, starting with the primary visual cortex and moving out. And we can find these visual areas in these experiments called retinotopic mapping experiments. So,
could you describe how that works and what those are?
Right. So for every one of our subjects in a different session, we run these experiments where we find out where the visual areas are using these mapping techniques. And once we find these areas in a given subject, we keep their phone number so we can bring them back and use them in subsequent experiments. So we don't have to do it all in one day. Mm-hmm. The way these experiments work is they take advantage of the way these receptive fields in the visual cortex are organized. So area V1, primary visual cortex, is organized in a map. It's basically kind of a reflection of the retina back here. So that if neurons that are neighboring each other in V1 have receptive fields, or places of the visual field that they like to respond to, near each other. So if I were to present a stimulus that, say, moves around in a systematic way in visual space, it's going to produce this wave of responses across the map in the primary visual cortex. And there's basically 2 dimensions to this map. One is how far eccentric the stimulus is from the center of fixation, and sort of the orthogonal dimension is sort of how radially they are, say, it's like the second hand of a clock. And so if I present a stimulus that slowly expands over time, it produces this particular wave of activity. And if I have a stimulus that slowly rotates around like a second hand of a clock, it produces a wave sort of in the 90-degree angle direction. And it turns out that primary visual cortex has this map. Area V2, which is split in half, lies right next to area V1, has a similar map, but it's kind of flipped inside. It's basically upside down from V1. And so the waves are in the opposite direction. V3, the next area, is in the same direction as V1, as though it's V2 flipped over. So what we can do is use these experiments where these stimuli rotating around slowly in time or expanding slowly in time and look at the directions of motion of these waves of activity and find out where V1, V2, V3, et cetera are.
Wait, so does every functional area in the visual cortex represent the entire visual field?
Kind of, right. So primary visual cortex on the left side of the brain represents the whole right side of the visual field and vice versa. And the upper half of V1 represents the lower half of the visual field and vice versa. V2 is interesting because it actually is 4 separate components. They're lying on either side of V1 on both the left and right side. Each of those represents 90 degrees of the visual field, one particular quadrant. Lower V2 represents the upper quadrant on the other side of the visual field. So there's actually, V2 is call it one area, but it's really 4 separate chunks of the brain, each representing each one of 4 quadrants.
And that continues all the way up to MT or higher order visual processing areas?
We think so. And of course, so the idea of a map is really defined in physiology by several factors, not just the retinotopic map, but also the functionality and the connections between these maps. All we have with this fMRI is just the retinotopic map. And so there's been a lot of research trying to figure out where these maps are in the human brain, how many they are, and how they relate to, say, monkey physiology, which have been measured much more carefully. And so there are now, we're up to more than a dozen different retinotopic maps we found in the human visual system that go all the way up into the parietal lobe or down into the ventral surface of the brain. And the number keeps going up.
Are there other modalities in which they're organized in a particular fashion?
That's a great question. I think there's a lot of vision research with fMRI, partly because the light is good there. We can measure these maps. Now when we get a brain response with fMRI, we know which visual area this response is coming from. Other types of experiments, if you're doing memory experiments or language experiments, we don't have these really nice predefined maps that we can measure functionally. In other sensory areas, it's not so easy. The exception, actually, it's like the visual system, is the somatosensory system. We have this nice somatosensory homunculus.
So getting back to the fMRI experiment, now that you've mapped all the different visual areas, how do you account for individual differences in the cortical, or in the cortex, or other areas of the brain?
Well, the nice thing about studying the visual system with fMRI is that because we have a separate map, we know where the visual areas are for each individual subject. We don't have to worry about individual differences so much, because then we do functional experiments, we just simply align the data with that subject's map, and we know which visual areas the responses are happening. But for experiments outside these nicely defined mapped areas, there's a lot of issues. Language, memory, prefrontal cortex, all these other areas. There are no standard techniques for finding where the dorsolateral prefrontal cortex is in an individual. So it's very difficult to average data across subjects, for example, because, like you mentioned, individual differences in the anatomy of the brain can cause all sorts of trouble with us.
Is there a standard mapping system that everyone relies on, like an average brain that you map all the other brains to?
Mm-hmm. There's a couple. The most standard one is called a Talerac map. It's a standard brain that has a particular size, and when you get functional data from another subject, you basically squash you distort your data set to fit this particular template. And this template has X, Y, Z coordinates. And so if you get a spot of activity in this particular box in this brain, you can look at this standard template and say, well, that looks like it must be in the dorsolateral prefrontal cortex. But the problem is the folds in gyri and sulci in an individual subject, they vary all over the place. And so you can be way off in terms of what part of the brain you're thinking about when you put these things in these Talerac coordinates.
Are there certain areas that differ more? Are there, like, what I mean by that, are there landmark areas that are consistent throughout people or participants?
So yeah, the standard brain has some very standard features. For example, you've got the precentral sulcus and obviously the midline of the brain, the 2 hemispheres. There's some very basic things that we can use as landmarks. But the subtle sulci and gyri, although they all have names, they vary a lot, and even in number across individuals. Primary visual cortex, for example, you'd think that would just be fixed in stone. They are different shapes. My primary visual cortex pretty much sits inside a fold called the calcarine sulcus in the back of the head. which is basically in the midline between the two hemispheres. My left V1, primary visual cortex, points upward, and my right one points horizontally. They're completely separate between the two hemispheres. And so you can imagine if you just average data across subjects blindly, you're going to really run into trouble about where you think your data is coming from.
That's really interesting. Why don't we continue this conversation in our next segment?